second half of the mitosis or cell growth notes. As you learned in the previous one, there is interphase. An interphase is the holding stage or the stage between two successive cell divisions. Successive cell divisions would mean back to back. So interphase is the phase that your cells are in almost all the time. That's when they're just kind of hanging out, being good old cells. So 90% of a cell's time in the normal cellular cycle may be spent in interphase, doing just what they're supposed to be doing. There are 3 phases to the interphase stage. Those would be G1, which we talked about on the previous video, which is growth, S, synthesis of DNA, and G2, which is a prep for mitosis. Once again, the 3 phases of interphase are G1, S, and G2, which was on the other video. And if we're looking at this as an entire cycle, you can see exactly what the interphase cell will look like. It's got the centrioles, it shows the nuclear envelope, it shows the chromatin, and you'll look on the inside. What you're going to notice is this is going to change considerably as we move through the cell cycle. Prophase. Prophase is the first and longest phase of mitosis. Cells are in this particular phase between 50 and 60% of the total time. Chromosomes are visible. Centrioles separate and move to the opposite side of the nucleus. A spindle can form, and the spindle is what's going to help to separate the chromosomes. The definition of a spindle is a fan-like microtubule, once again, that helps to separate the chromosomes. Chromosomes attach to these spindle fibers at what's called the centromere of each chromatid. And in the notes previously, we talked about the parts and pieces to the chromosome. We talked about the 2 sister chromatids that are identical and how they're attached at the centromere. At the end of prophase, chromosomes coil tightly, the nucleolus, which is of course inside the nucleus, disappears, and the nuclear envelope breaks down. Here is then a picture, and this is extremely important to know what these pictures look like, because you're going to have to identify them. Once again, you've got the centrioles. They kind of look like this, and they have like tubules in there. That's what you can see the spindle fibers that are forming. When you see this dotted line, that means that the nuclear envelope or nuclear membrane is breaking down. That's what the dotted line stands for. You can see the chromosomes, which are paired sister chromatids. And then the centromere is that little space that attaches them in the middle. So that's prophase. Metaphase. During metaphase, chromosomes line up across the center. So if you remember, metaphase equals middle. And I suppose I should have backed up. Pro means first. That's how you know that that's the first phase of mitosis. Pro means first. So back to this. During metaphase, chromosomes line up across the center of the cell or the middle. It only lasts for a few minutes, so it's extremely short. Microtubules connect the centromere to the poles of the spindle. And as you can see in this particular picture, you can see all the chromosomes are lined up. This would be the cell membrane around here. That's the cell membrane. You can see the little spindle fibers that are coming, and your centrioles are going to be down here with your chromosomes in the middle. And once again, here is another picture. And you can see where they're lined up in the middle. Once again, we've got our centrioles, we've got the spindle fibers, and they're right down the center of that cell. Next phase is anaphase. And anaphase starts right when those chromosomes decide to pull away from the center. So during anaphase, the centromeres that join the sister chromatids split, allowing the sister chromatids to separate and become individual chromosomes. So as you can see on this picture down here, this is the middle, and these are the individual ones. They're now starting to pull away. And here's where they were joined together. They were joined here. They're starting to pull towards each of the ends down here. Chromosomes move until they have separated into two groups near the poles of the spindle. And anaphase ends when the chromosomes stop moving. So once those chromosomes reach the end point down here, once they have pulled all the way back to the ends, would signal the end of anaphase. And once again, just to clarify, the second that they move away from the middle, that's when anaphase begins. And now each of these here are individual chromosomes. They used to be attached together, then they were sister chromatids, so they were paired. Now they're back to being individual again. Telophase, and the way that you can remember this one is telo, you're going to end up with 2 cells. So during telophase, the chromosomes begin to disperse into a tangle of dense material. And you see in this picture down here where they're now starting to bunch up. They're all starting to bunch up. You can still tell that they're individual strands, but they're now starting to form a mass. A nuclear envelope reforms around each cluster of chromosomes. So the nuclear envelope comes back. The spindle breaks apart. A nucleolus becomes visible in each daughter nucleus, making mitosis almost complete. So if we're looking on our picture here, telophase, the way I remember it, is 2 because you're ending up almost with 2 identical cells. You can see by that dotted line, the dotted line means the nuclear envelope is reforming around the genetic material. And you can see by looking on the inside as compared to what was up here, here you just have your individual or your single chromosomes. Up here, they are paired. The last phase after we've gone through mitosis is called cytokinesis. And of course, the word cyto means cell. Cyto means cell. As a result of mitosis, 2 nuclei are now formed, usually within the cytoplasm of a single cell. The final step is the division of the cytoplasm. So, cytokinesis is the division of the cytoplasm. That's what cytokinesis means. Cytokinesis usually occurs at the same time as telophase. and this is important to remember. Cytokinesis usually occurs at the same time as telophase. In animal cells, the cell membrane is drawn inward until the cytoplasm is pinched into two nearly equal parts. And if we're looking at this particular cell here, you can see where that cell membrane is starting to reform around each of those cells. And this part where it kind of comes in and then goes back out, that is called a cleavage furrow. So there we have the final picture where at the end of cell division, you are going to have two identical cells that have the same, so both of these, you have two identical cells. They have the same genetic material inside of them. And these two cells look similar to that original cell that you started with before you started to go through the whole cell cycle. Once again, interphase is the resting phase, prophase is first, metaphase is middle, anaphase is where they pull away, and telophase means two. Cytokinesis is split of the cytoplasm. And this concludes the notes on mitosis and cell division.